The main goal of this experiment is to obtain a glucose solution through the enzymatic hydrolysis of pretreated aspen wood chips using specialized enzymes, called cellulases. The second goal is to estimate the yield of glucose from this reaction.
For this experiment, you will need the following pieces of equipment: the 10 to 100 microliter pipette, the 100 to 1000 microliter pipette, pipette tips, 2 50 milliliter Erlenmeyer flasks, a hot plate, a lab notebook and pencil, the pressure cooker, tinfoil, parafilm, wax paper, a digital thermometer, a scale, gloves, safety glasses, and a glucose meter with test strips. You will need the following chemicals: pretreated aspen wood chips, raw aspen wood chips, distilled water, sodium citrate buffer, which has a pH of 4.5, the antibiotic solutions tetracycline and cyclohexamide, and the enzymes Accelerase 1500 and Accelerase BG.
Make sure to keep an organized lab notebook. Record all dates, procedures, and results. Wear safety glasses, long pants, and closed-toed shoes. Also wear gloves to avoid contact with chemicals. Ethanol is highly flammable and must be kept away from ignition sources. Be cautious with the pressure cooker and the hot plate to avoid burns and electrical shocks. Handle glass beakers with care to prevent breakage.
The wood chips that you have received have already been pre-treated with heat and dilute sulfuric acid. During pretreatment, the hemicellulose fraction of the wood was reacted away by the acid catalyst to form a sugar solution containing 5 and 6 carbon sugars. This reaction has the secondary effect of opening up the structure in the wood so that the cellulose can be accessed by the enzymes. During enzymatic hydrolysis, the process that we will start now, enzymes convert the cellulose in the biomass into glucose. This reaction will occur in the presence of antibiotics in order to preserve the glucose in solution from microbial attack.
Using the scale, measure out 1 gram of moist, pre-treated aspen wood chips and place them into a 50 milliliter Erlenmeyer flask. Make sure to zero the scale so as not to include the mass of the weighing dish in your measurement.
A control flask containing all components except the wood chips should be made. The purpose of the control is to isolate the effects of the independent variable, the aspen wood chips, from the other variables and to account for any glucose present in the other solution components, like the enzymes. A second control containing 0.3 grams of raw aspen wood chips instead of the pre-treated wood chips can also be made to show the importance of dilute acid pretreatment on the effectiveness of the enzymes. Using a graduated cylinder, add 19 milliliters of distilled water to both flasks. Make sure to measure the bottom of the meniscus.
Now we will teach you how to use a pipette. First, unlock the knob on top of the pipette. Then, turn the knob until you reach your desired volume. Remember, the volume of the pipette is given in microliters. To convert to milliliters, divide by 1000. After adjusting to the correct volume, put a plastic tip on your pipette, press the knob on top of the pipette down to the first stop, and place the pipette in the solution. Release the button slowly to pull the liquid into the pipette tip. No solution is to touch the bottom of the pipette housing. Then, push the button down all the way to the second stop in order to release the liquid into the desired container. Remember to change out pipette tips when using different chemicals to avoid cross-contamination.
Using the 100 microliter to 1000 microliter pipette, add 1 milliliter of sodium citrate buffer to both flasks.
Add 80 microliters of tetracycline, an antibiotic against bacteria, to both flasks. This should be done using the 10 microliter to 100 microliter pipette. Remember to wear gloves to protect yourself from having solution touch your skin. The chemicals in this experiment are not very hazardous, but we want to teach good laboratory practice and also limit overexposure to antibiotics. Using the 10 microliter to 100 microliter pipette, add 60 microliters of cyclohexamide, an antibiotic against yeast, to both flasks.
Put a rubber stopper on each flask. Cover the stopper with parafilm, then tinfoil.
Pour 1 liter of water into the bottom of the pressure cooker. Place the pressure cooker on the hot plate and place the flasks in the pressure cooker. Using a thermometer, monitor the temperature of the water and bring the flasks to 50 degrees Celsius. Do not place the lid on the pressure cooker during this process. The flasks should be kept at this temperature for 1 hour, then removed from the pressure cooker. The pressure cooker should remain on the hot plate, as the flasks will be placed in it again later. Remember to wear protective gloves, as the pressure cooker and flasks will be hot.
For the next step, the dry weight of the biomass must be determined. The pre-treated aspen wood chips are about 30% dry mass, so 1 gram of wet wood chips yields 0.3 grams of dry wood chips.
The following enzymes should be added to the flasks. One enzyme is called Accelerase 1500. This is actually a solution of more than one enzyme acting together to break down the cellulose. 250 microliters of this enzyme should be added for each gram of dry biomass. This means that for our 0.3 grams of dry biomass, 75 microliters of Accelerase 1500 should be added. This should be done using the 10 microliter to 100 microliter pipette. Our other enzyme is called Accelerase BG. 90 microliters of this enzyme should be added for each gram of dry biomass. Since we have 0.3 grams of dry biomass, 27 microliters of Accelerase BG should be added, using the 10 microliter to 100 microliter pipette. Accelerase BG takes the reaction to completion by producing glucose from the cellulose breakdown products.
The flasks should be kept at a constant 50 degrees Celsius in the pressure cooker. The enzymes need a warm environment to convert the cellulose into glucose. Using the 10 microliter to 100 microliter pipette, a 10 microliter sample should be removed from each flask every 24 hours, starting at time 0. Release the sample onto a piece of wax paper on the lab bench top and allow 30 seconds or so for it to come to room temperature. These samples will be tested for glucose to determine how much is being produced over time.
Use the blood glucose meter and test strips to determine glucose concentrations in the 10 microliter samples taken from each flask. To turn on the glucose meter, press the center button. Once a blinking arrow appears on the screen, insert the test strip into the bottom. Once a blinking droplet appears, place the test strip into the sample on the wax paper. Record the reading from the glucose meter in your lab notebook.
Make standard glucose solutions with concentrations of 1, 2, 4, and 5 grams per liter. Standard solutions are prepared by combining glucose with the background solution containing 19 milliliters distilled water, 1 milliliter sodium citrate buffer, 80 microliters of tetracycline, and 60 microliters of cyclohexamide. The purpose of using the background solution to mix the glucose standards is to account for any glucose already present. The glucose standards allow us to make a calibration curve in order to more accurately measure the glucose converted during hydrolysis. For the 5 grams per liter solution, mix 0.05 grams glucose with 10 milliliters of background solution. A scale and graduated cylinder should be used for measurement. All other solutions will be made by diluting this 5 grams per liter solution. For all solutions, the 100 microliter to 1000 microliter pipette should be used. For the 4 gram per liter solution, mix 800 microliters of 5 gram per liter solution with 200 microliters of the background solution. The 2 grams per liter solution should be made by mixing 400 microliters of 5 gram per liter solution with 600 microliters of the background solution. For the 1 gram per liter solution, mix 200 microliters of 5 gram per liter solution with 800 microliters of the background solution. Measure each standard solution 2 times with the glucose meter. 10 microliter samples should be taken for each test. Use the 10 microliter to 100 microliter pipette. Average the results from each solution. Record your results since these numbers will be needed to compare to glucose readings of the biomass solution and create a calibration conversion.
You will need to graph your calibration curve on Excel. The blood glucose meter determines the glucose levels in a solution by measuring the rate of the reaction that occurs when the enzymes on the test strip consume the glucose in the solution. However, when the solutions have concentrated glucose levels, the enzymes on the test strips become saturated. They cannot process the glucose fast enough, and the reading becomes inaccurate. This typically occurs at concentrations of 4 to 5 grams of glucose per liter of solution. This is also where your graph typically flatlines. When making a best fit line for your calibration curve, choose the regression line with the highest R-squared value. It will most likely be a polynomial regression line. Make sure to record the regression equation. With it, you will be able to convert the glucose concentrations recorded from the hydrolysis into real concentrations.
We will now analyze the results to determine how much of the biomass was converted into glucose. Keep in mind that the dry mass of the biomass is about 30% of the wet mass. Of the dry mass, only about 50% is cellulose. Cellulose is a substance which can be converted into glucose. With that information, you should be able to determine your theoretical glucose yield. With 0.3 grams of dry wood chips containing about 50% cellulose, your theoretical yield should be around 0.15 grams of glucose. To determine your actual glucose yields, first determine the total volume of solution. Add the volumes of all of the components. It should be 20.242 milliliters when you add together the 19 milliliters of distilled water, 1 milliliter of sodium citrate buffer, 80 microliters of tetracycline, 60 microliters of cyclohexamide, 75 microliters of accelerase 1500, and 27 microliters of accelerase BG.
To calibrate our results, we took the meter reading and converted it into actual glucose concentrations using the regression equation from our calibration curve. Here are the results from our control solution, unpretreated wood chips and pretreated wood chips. Notice that the control solution has some background glucose in it. Also, the difference in glucose concentrations between the pretreated and unpretreated wood chips shows the necessity for pretreatment. Our wood chips only hydrolyzed for about 24 hours, so the glucose concentrations aren't as high as they would be if the enzymes had more time to work.
Since the control solution contains some glucose, we must subtract the background reading from the final unpretreated reading to get the net glucose concentration in the solution.
Use the final glucose concentration from the meter reading and the volume of the solution to determine the mass of the glucose in the solution. The calculation will be as follows, where everything on the top row is multiplied together and then divided by everything on the bottom row.
Here is the calculation for our glucose yield after 24 hours. Use the final mass of the glucose and the theoretical yield to determine the percent yield. This should be done by dividing the actual yield by the theoretical yield and then multiplying the result by 100.
Here is the calculation for our percent yield when the wood chips are given around 3 days to hydrolyze. The percent yield should be more like 60%. This experiment may be done by teams of students. For example, one team could make the control solutions, another team could make the solution with the pre-treated wood chips, another the solution with the raw wood chips, while one team preheated the pressure cooker, while another group made and tested the glucose standard solutions.